Chapter 12 Mineral Nutrition. The basic needs of all living organisms are essentially the same. They require macromolecules such as carbohydrates, proteins, and fats, and water and minerals for their growth and development. This chapter focuses mainly on inorganic nutrient nutrition, wherein we will study the methods to identify elements essential to growth and development of plants and the criteria for establishing the essentiality. We will also study the role of essential elements, their major deficiency symptoms, and the mechanism of absorption of these essential elements. This chapter also introduces you briefly to the significance of the mechanism of biological nitrogen fixation. 12.1 Methods to study new requirements of plants. In 1860, Julian von Sachs, a prominent German botanist, demonstrated for the first time that plants could be grown to maturity in a defined nutrient solution in the complete absence of soil. This technique of growing plants in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrients essential for plants. The essence of all these methods involves the nurture of plants in a solidly defined mineral solution. These methods require purified water and mineral nutrient salts. Can you explain why is this so essential? After a series of experiments in which the roots of plants were immersed in a nutrient solutions and wherein an element was added or substituted or removed or given in varied concentration, a mineral solution suitable for plant growth was obtained. By this method, essential elements were identified and the deficiency symptoms discovered. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomato, seedless cucumber, and lettuce. It must be emphasized that the nutrient solution must be adequately aerated to obtain the optimum growth. What would happen if solutions were poorly aerated? Diagrammatic use of hydroponic technique is given in Figure 12.1 and 12.2. Figure 12.1 diagram of a typical setup for nutrient solution culture. Funnel for adding water and nutrient solution, aerating tube, cotton. Figure 12.2 Hydroponic plant production. Plants are grown in a tube or a tube placed on a slant incline. A pump circulates a nutrient solution from a reservoir to the elevated end of the tube. The solution flows down the tube and returns to the reservoir due to gravity. Insert shows a plant uses the roots continuously bathed in aerated nutrient solution. The arrows indicates the direction of the flow. Nutrient solution in the pump. 12.2 Essential mineral elements. Most of the minerals present in the soil can enter plants through roots. In fact, more than 60 elements of the 105 discovered so far are found in different plants. Some plant species accumulate selenium, some other gold, some others gold, while some plants grow nuclear pesticides take up radioactive strontium. There are techniques that are able to detect the minerals even at a very low concentration that is 10 to -8 gram per ml. The question is whether all the diverse mineral elements present in the plant, for example, gold and selenium as mentioned above, are really necessary for plants. How do we decide what is essential for plants and what is not? 12.2.1 Criteria for essentiality. Criteria for essentiality of an element are given below. A. The element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plants do not complete their life cycle or send the seeds. B. The requirement of the element must be specific and not replaceable by another element. In other words, deficiency of any one element cannot be met by supplying some other element. C. The element must be directly involved in the metabolism of the plant. Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirements. First, macronutrients and second, micronutrients. Micronutrients are generally present in plant tissues in large amounts in excess, that is 10 millimole per kg of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Of these, carbon, hydrogen, oxygen are mainly obtained from CO2 and H2O, while the others are absorbed from the soil as mineral nutrition. Micronutrients or trace elements are located in very small amounts, less than 10 millimole per kg of dry matter. These include iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to the 17 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt, selenium. They are required by higher plants. Essential elements can also be grouped into four broad categories on the basis of their diverse functions. These categories are: first, essential elements are as components of biomolecules and structural elements of cells, for example, carbon, nitrogen, oxygen, and nitrogen. Second, essential elements that are components of energy-related chemical compounds in plants, for example, magnesium in chlorophyll, and phosphorus in ATP. Third, essential elements that activate or inhibit enzymes. For example, Mg2 plus is an activator for both ribulose, phosphate, carboxylase, oxygenase, and phosphoenol pyruvate carboxylase, both of which are critical enzymes in photosynthetic or carbon fixation. Zn2 plus is an activator of alcohol dehydrogenase and MO, that is molybdenum of hydrogenase during nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathways you have studied earlier. 4. Some essential elements can alter the transport potential of a cell. Potential plays an important role in the opening and closing of the stomata. You may recall the role of the minerals and solutes in determining the water potential of a cell. 12.2.2 Role of macro and micronutrients. Essential elements perform several functions. They participate in various metabolic processes in the plant cells, such as permeability of the cell membrane, maintenance of osmotic concentration of cell sap, electron transport system, buffering action, enzymatic activity, and act as major constituents of macromolecules and coenzymes. Various forms and functions of essential nutrient elements are given below. Nitrogen. This is the essential nutrient element required by plants in the greatest amount. It is absorbed mainly as NO3-, though some are also taken up as NO2- or NH4+. Nitrogen is required by all the parts of the plant, particularly the meristematic tissues and the metabolically active cells. Nitrogen is one of the major constituents of the proteins, nucleic acids, vitamins and hormones. Phosphorus is absorbed by the plant from the soil in the form of phosphate ions, either as H2PO4- or HPO42-. Phosphorus is a constituent of cell membranes, certain proteins or nucleic acids and nucleotides, and is required for all the phosphorylation reactions. Potassium, it is absorbed as potassium ion. Potassium, it is absorbed as potassium ion K+. In plants, this is required in more abundant quantities in the meristematic tissues, buds, leaves, and root tips. Potassium helps to maintain an anion-cation balance in cells and is involved in protein synthesis, opening and closing of the stomata, activation of enzymes, and in the maintenance of the fragility of the cells. Calcium. Plant absorbs calcium from the soil in the form of calcium and Ca2+. Calcium is required by the meristematic and the differentiating tissues. During cell division, it is used in the synthesis of cell wall, particularly as a calcium peptide in the middle lamina. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in the normal functioning of the cell membranes. It activates certain enzymes and plays an important role in their regulating metabolic activities. Magnesium is observed by the plants in the form of diamond and Mg2+. It activates the enzyme of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plants contain sulfur in the form of sulfate SO4 2-. Sulfur is present in two amino acids, cysteine and methionine, and the main constituent of the several coenzymes: vitamins, thiamine, biotin, coenzyme A, and ferredoxin. Iron. Plants obtain iron in the form of ferric ions Fa3+. It is required in larger amounts in comparison to the other micronutrients. It is an important constituent of proteins involved in the transfer of electrons like ferredoxin and cytochromes. It is reversibly oxidized from Fe2+ to Fe3+ during electron transfer. It activates catalase enzyme and is essential for the formation of chlorophyll. Manganese is absorbed in the form of manganese ions Mn2+. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. So, let's define function of manganese is in the splitting of the water to liberate oxygen during photosynthesis. Zinc. Plants obtain zinc as Zn2+ ions. It activates various enzymes, especially carboxylases. It is also needed in the synthesis of oxygen. Copper. It is absorbed as cupric ion Cu2+. It is essential for the overall metabolism in plants. Like iron, it is associated with certain enzymes involved in reduction and is reversibly oxidized from Cu+ to Cu2+. Boron. It is absorbed as BO3- or B4O72-. Boron is required for uptake and utilization of Cu2+. Membrane functioning, ozone germination, cell elongation, cell differentiation, and carbohydrate transportation. Molybdenum plants obtain it in the form of molybdate ions MoO2+. It is a component of several enzymes including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism. Fluorine is absorbed in the form of chlorine, chloride anion, Cl- along with the Na+ and K+. It helps in determining the soluble concentration and the anion-cation balance in cells. It is essential for the water splitting reaction and photosynthesis, a reaction that leads to oxygen evolution. 12.2.3 Deficiency Symptoms of essential elements. Whenever the supply of an essential element becomes limited, plant growth is retarded. The concentration of the essential element below which plant growth is retarded is termed as the critical concentration. The element is said to be deficient when present below the critical concentration. Since each element has one or more specific structural or functional role in plants, in the absence of any particular element, plants show certain morphological changes. These morphological changes are indicative of certain element deficiencies and are called deficiency symptoms. Deficiency symptoms vary from element to element and they disappear when the deficient mineral nutrient is provided to the plant. However, if deprivation continues, it may eventually lead to the death of the plant. The parts of the plant that show the deficiency symptoms also depend on the mobility of the element in the plant. For elements that are actively mobilized within the plants and exported to young developing tissues, the deficiency symptoms tend to appear first in the older tissue. For example, the deficiency symptoms of nitrogen, potassium, and magnesium are visible first in the senescent leaves. And the older leaves biomolecules containing these elements are broken down, making these elements available for mobilizing to younger leaves. The deficiency symptoms tend to appear first in the young tissues whenever the elements are relatively mobile and are not transported out of the mature organs. For example, elements like sulfur and calcium are a part of the structural component of the cell and hence are not easily released. This aspect of mineral nutrition of plant is of a great significance and importance to agriculture and horticulture. The kind of deficiency symptoms is shown in plants like chlorosis, necrosis, rapid plant growth, premature fall of leaves and buds, and inhibition of cell division. Chlorosis is the loss of chlorophyll leading to yellowing in leaves. The symptom is caused by the deficiency of elements nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc, and molybdenum. Likewise, necrosis or depth of tissue, particularly leaf tissue, is due to the deficiency of Ca, Mg, Cu, K, lack or low level of N, King, S, molybdenum, causes an inhibition of cell division. Some elements like N, S, MO, delay flowering if their concentration in plants is low. You can see from the above that the deficiency of any element can cause multiple symptoms and that the same symptoms may be caused by the deficiency of one or of several different elements. Hence, to identify the deficient element, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard tables. We must be aware that different plants also respond differently to the deficiency of the same element. 12.2.4 Toxicity of macronutrients. The requirement of micronutrients is always in low amounts while they are moderate degrees causes the deficiency symptoms and a moderate increase causes toxicity. In other words, there is a narrow range of concentration at which the elements are optimal. Any mineral iron concentration in tissues that reduces the dry rate of tissue by about 10% is considered toxic. Such critical concentrations vary widely among different macronutrients. The toxicity symptoms are difficult to identify. Toxicity levels for any element also vary for different plants. Many a times excess of an element may inhibit the uptake of another element. For example, the prominent symptom of manganese toxicity is the appearance of brown spots surrounded by the chlorotic veins. It is important to know that manganese competes with iron and magnesium for uptake and with magnesium for binding with enzymes. Manganese also inhibits calcium translocation in short effects. Therefore, excess of manganese may in fact produce deficiency in induce deficiency of iron, magnesium, and calcium. Thus, what appears as symptoms of manganese toxicity may actually be the deficiency symptoms of iron, magnesium, and calcium. Can this knowledge be of some importance to a farmer, a gardener, or even for you in your kitchen garden? 12.3 Mechanism of absorption of elements. Much of the studies on mechanism of absorption of elements by plants has been carried out in isolated cells, tissues, or organs. These studies reveal that the process of absorption can be demarcated into two main phases. In the first phase, the initial rapid uptake of ions into the free space or outer space of cells, the protoplasm is passive. In the second phase of uptake, the ions are taken in slowly into the inner space, the interest of the cells. The passive movement of ions into the protoplasm usually occurs through ion channels. The transmembrane proteins that function as selective doors. On the other hand, the entry or exit of ions to and from the protoplasm requires expenditure of metabolic energy, which is an active process. The movement of ions is usually called flux. The inward movement in the cells is influx and the outward movement efflux. You have read the aspects of mineral nutrient uptake and transportation at the reactor level. 12.4 Translocation of solutes. Mineral salts are translocated to The item along with the assembling stream of water, which is pulled up through the Prana transpiration and pull. Analysis of the item's have shows the presence of mineral salts in it. Proofs of the radioisotope isotopes of mineral elements also substantiate the view that they are transported through the xylem. We have already discussed the movement of water in xylem in chapter 11. We confine soil as a reservoir of essential elements. Majority of the nutrients that are essential for the growth and development of plants become available for the roots due to the weathering and breakdown of rocks. These processes enrich the soil with the salt, ions, and organic salt. Since they are derived from the rock mineral, their role in plant nutrition is referred to as mineral nutrition. Soil consists of a wide variety of substances. Soil not only supplies the minerals but also harbors nitrogen-fixing bacteria. Other microbes also water supplies air to the roots and acts as a matrix that stabilizes the plant. Since deficiency of essential mineral affects the crop yield, there is often a need for supplying them through fertilizers. Both macronutrients NPK, etc. and micronutrients Cu, Zn, Fe, Mn, etc. form components of fertilizers that are applied as per need. 12.6 Metabolism of nitrogen. 12.6.1 Nitrogen cycle. Apart from carbon, hydrogen and oxygen, nitrogen is the most prevalent element in living organisms. Nitrogen as a constituent of amino acids, proteins, hormones, chlorophylls, and many of the vitamins transported to microbes for the limited nitrogen that is available in soil. Because nitrogen is a limited nutrient for both the natural and agricultural ecosystem. Nitrogen exists as two nitrogen atoms joined by a ring from triple covalent bond in triple bond N. The process of conversion of nitrogen into ammonia is termed as nitrogen fixation. In nature, light and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxide, NO2, and NO2. Industrial combustion, forest fires, automobile exhaust, and power generating station are also sources of atmospheric nitrogen oxides. Decomposition of organic nitrogen of red plants and animals into ammonia is called ammonification. Some of this ammonia volatilizes and reacts to the atmosphere but more strict. It is converted into nitrate by soil bacteria in the following steps: 2NH3 plus CO2 plus 2NO2 minus plus 2H plus 2H2O, 2NO2 minus plus O2 plus 2NO3. Ammonia is the first oxidized to nitrite by the bacterial also known as or nitrococos. The nitrite is further oxidized to nitrate by the help of the bacterial nitrobacter. These steps are called nitrification. These nitrifying bacteria came out of rocks. The nitrate in this form is absorbed by plants and is transported to the leaves. In leaves, it is released to form ammonia that finally forms the amine group of amino acids. Amine group of amino acids. Nitrate plus method is also reduced to nitrogen by the process of denitrification. Denitrification is carried by the bacteria Pseudomonas and Lactobacillus. Figure 12.3 the nitrogen cycle, showing relationship between the three main nitrogen pools: atmospheric, soil, and biomass. Nitrogen, atmospheric N2, biological N2 fixation, industrial interfixation, electrical interfixation, biological N2 fixation and industrial interfixation gives NH3, that gives NO2 minus, soil N2, that gives NO3 minus. Electrical nitrogen fixation and denitrification gives that atmospheric nitrogen, NO3 minus of the plant biomass, and to animal biomass, and then to decaying biomass, and then ammonification gives NH3, and the cycle continues. 12.6.2 Biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form of N2 available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Production of nitrogen to ammonia by living organisms is called biological nitrogen fixation. Enzyme nitrogenase, which is capable of nitrogen production, is present exclusively in the prokaryotes. Such microbes are called nitrogen fixers. N3 in the process of nitrogen is NH3. The nitrogen fixing microbes could be free living symbiotic. Examples of free living nitrogen fixing aerobic microbes are Azotobacter and Bacillomycia, Pyrrhodomus pyralium is anaerobic and Bacillus free living. In addition, a number of cyanobacteria such as Amebina and Hostrop are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixing associations are known. The most prominent among them is a legume-bacteria relationship. Species of roll-shaped rhizobium has such a relationship with the roots of the several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. The most common association on roots is as nodules. These nodules are small nodules on the roots. The micropharangia also produces nitrogen-fixing nodules on the roots of non-leguminous plants, for example, almonds. Both rhizobia and pharangia free living on soil, but as symbiont can fix atmospheric nitrogen upload any one plant of a common pulse just before flowering. You will see near spherical outgrowths on the roots. These are nodules. However, through them you will notice that the central portion is red or pink. What makes the nodule pink? This is due to the presence of leguminous hemoglobin or leghemoglobin. Nodule formation. Nodule formation involves the sequence of multiple interactions between rhizobium and roots of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobia multiply and colonize the surroundings of the roots and get attached to the epidermal and root hair cells. The root hair curls of the bacteria invade the root hair. An infection thread is produced carrying the bacteria into the cortex of the cell, cortex of the root, where they initiate the nodule formation in the cortex of the root. Then the bacteria produced from the thread into the cells, which leads to the differentiation of the specialized nitrogen-fixing cells. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients. These events are depicted in figure 12.4. The nodule contains all the necessary biochemical components such as the enzyme nitrogenase and leghemoglobin. The enzyme nitrogenase is MOFE protein and catalyzes the conversion of atmospheric nitrogen to ammonia, figure 12.5, the first stable product of nitrogen fixation. Figure 12.4, development of root nodules is where the rhizobium bacteria contact the susceptible root hair divides near it. Soil particles, root hair, bacteria. B. Successful infection of the root hair was susceptible to bacteria. C. Infected thread carries the bacteria to the inner cortex. The bacteria get modified into rod-shaped bacterioids and causes inner cortical and pericycle cells to divide. Division and growth of cortical pericycle cells leads to nodule formation. C. Infection thread containing bacteria. Inner cortex and pericycle cells under division. D. A mature nodule is complete with vascular tissues continuous with growth of the root. Mature nodule. The reaction is as follows. N2 plus 8 electrons plus 8 H plus N plus 16 ATP equals to 2 NH3 plus H2 plus 16 ADP plus 16 IP. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic conditions and nodules have adaptations that ensure that the enzyme is protected from the oxygen. To protect these enzymes, the nodule contains an oxygen scavenger called leghemoglobin. It is interesting to note that these microbes live as aerobic under free living conditions when nitrogenase is not operational. But during nitrogen fixing event, they become anaerobic, thus protecting the nitrogenous inside. You must have noticed in the above reaction that the ammonia synthesis of nitrogenase requires a very high input of energy, 8 ATP for each NH3 produced. Energy required thus is obtained from the respiration of the host cell. Rate of ammonia at physiological pH. Ammonia is protonated to form NH4 plus ammonium ion, while most of the plants can stimulate and nitrate as well as ammonium ions. Later is quite toxic to plants and hence cannot accumulate in them. Let us now see how the NH4 plus is used to synthesize amino acids in lungs. These are two main ways in which this can take place. First, productive amination. In this process, ammonia reacts with alpha ketoglutaric acid and forms glutamic acid as indicated in the equation given below. Alpha ketoglutaric acid plus NH4 plus NADPH in the presence of glutamate and dehydrogenase gives glutamate plus H2O plus NADP. Second, transamination involves the transfer of amino group from one amino acid to the keto group of a keto acid. Glutamic acid is the main amino acid from which the transfer of NH2 in the amino group takes place, and the other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reactions. For example, R1CH NH3 plus COO minus amino donor plus R2C double bond O COO minus amino acceptor in the reversal process gives R1C double bond O COO minus plus R2CH NH3 plus COO minus. The two most important amides, asparagine and glutamine, found in plants are a structural part of proteins. They are formed from two amino acids, namely aspartic acid and glutamic acid, respectively by addition of another amino group to which the hydroxyl part of the acid is replaced by another NH2 minus radical. Since amides contain more nitrogen than the amino acids, They are transported to other parts of the plant via xylem vessels. In addition, along with the transpiration stream, the nodules of the some plants, for example, soybean, export the fixed nitrogen as ureas. These compounds also have a particularly high nitrogen to carbon ratio. Figure 12.5 steps of conversion of atmospheric nitrogen to ammonia by nitrogen is enzyme complex found in nitrogen fixing bacteria. Enzyme nitrogen is binding of substrate nitrogen gas into entropy bond and reduction double bond and mismatch. Again reduction and N H plus H and reduction NH3 and H3 3 nitrogen is and bind another molecular into product ammonia NH3 release of products. Summary from plants obtain the inorganic nutrients from air, water and soil. Plants absorb a wide variety of mineral elements. Not all the mineral elements that they absorb are required by plants. Out of more than 105 elements of discovered elements of organics and 21 are essential beneficial for novel plant growth and development. Elements required in large quantities are called macronutrients, while those required in less quantities are in trace part of those micronutrients. These elements are either essential constituents of proteins, carbohydrates, fats, and nucleic acids, and or take a part in various metabolic processes. Deficiency of each of these essential elements may lead to the symptoms called deficiency symptoms. Chlorosis, necrosis, and impaired division, etc. are some common deficiency symptoms. Once observed, minerals chlorose by either passive or active processes, and they are carried towards the parts of the organism through xylem along with the water transport. Nitrogen is very essential for the sustenance of plant. Plants cannot use the atmospheric nitrogen directly, but some of the plants in association into fixing bacteria, especially groups of legumes, can fix this atmospheric nitrogen into the biologically usable forms. Nitrogen fixation requires a strong reducing agent and energy in the form of ATP. N2 fixation is accomplished with the help of nitrogen fixing microbes, mainly rhizobium enzyme nitrogenase, which plays an important role in biological fixation. Is very sensitive to oxygen. Most of the processes takes place in the anaerobic environment. The energy it will be required is provided with the respiration of the host cell of the new produced following into fixation is incorporated into the amino acid as the amino group.